Hello. I think that the last video may have seemed like we covered quite a bit of material, and that can get sometimes a little overwhelming. But that was really just some general concepts so that we can understand everything that we will discuss in this video. So why don't we get started right away looking at this cellular division process known as mitosis. And you might remember this slide from our first video. So in this video, we will be looking at the cell division that takes place everywhere else in the body, but not the production of sex cells. So it is going to be every other cell, but not sperm and egg cells. So as we are ready to start mitosis, we have this cell where we can nicely see the nucleus and the nuclear pores. And inside there will be our DNA, which we will end up condensing into a structure that we saw before, a chromosome. Let's look at the first stage of the mitosis. During prophase, the nuclear envelope, which we just saw, disappears. You will see that the chromosomes are all over the place here. In addition to that, we will see that the centrosomes, which were these tubular pair structures that we talked on our review of the cell parts, these appear close to the nucleus. And from these also a spindle forms. Spindle is this bunch of protein fibers within the cytoplasm, and these will help with pulling the chromosomes to the right sides where they need to be. Now we move on to the second phase of the mitosis. It's called metaphase. During metaphase, chromosomes line up on an equatorial plane. So what it means is that they arrange themselves in a straight line between the centrosomes. The third stage is called anaphase. During this stage, the centrosomes and the spindle pull the chromosomes apart, cutting them into chromatid sticks, each of which moves to the one end of the cell. And it is noteworthy at this part to emphasize that the DNA content of each chromatid pair that earlier formed a chromosome together, well, those are identical. In addition to this, there is something else that we start to see. In an animal cell, the cell wall will start to pinch the two future cells apart. This is called cleavage starting to form. In plant cells, the cell starts to build a new wall, which will separate the resulting two cells. And the final stage, this is called teleophase. What happens here is that the cell has now divided into two. And we also notice that the nucleus and nuclear envelope reappears. So comes back. So here we see a summary of these phases of the mitosis. Please make sure to know what happens in each of these stages as the cell division process progresses through the mitosis. And there are many just as good summaries available if you feel that seeing it in a different format would help. And I think that I will be posting this to your study materials in case you want to have a closer look. To wrap up this video, I would like to say just a few words about different ways how the cell cycle can be regulated. So the first one that we will look at is the external stimulation. And this can be due to, for example, with growth factors or hormones. So various chemical messengers in the body can promote or trigger changes in the cells. So that would be how cell cycle, so the process of cell growing and then dividing into two, is stimulated. Let's have a look at how cell cycle process can be inhibited. So the opposite from what we saw earlier. One example of this, and the one that I have picked here, would be when cells grow in a closed space. Say, in this example, in a petri dish, they will keep growing happily until they reach a boundary, like the sides of the petri dish. This physical barrier will then work as a stop signal to the cell division, so to inhibit the cell cycle progress. And the final example that I have picked here relates to the fact that we already know. I am sure that we all agree that we can make a copy of the DNA and then a copy of that copy, and so on and so on. And to make this simpler to explain, I have changed these DNA strands that we see here to look like they would have been straightened out into a line, okay? And as we remember, not all of the DNA contained material that coded for genes. Much of it is just filler, if you wish to use that word. So on this figure, I have the genes marked with orange. But you might remember that whenever we made a copy of the original, the resulting copy is not quite as good quality as the first one. So just like taking a photocopy of a photocopy, the quality starts to decrease with each further copy. So this is also true for the copies of the DNA. For example, errors from previous copies, mutations, and so on, they all get also copied. So the mistakes start to accumulate. Well, this would not be good, right? Well, the evolution has designed a rather clever way to avoid this. Each DNA strand can be copied only so many times before we reach a signal embedded in the DNA that tells no more copies can be made. And on this graphical explanation, that signal will be the green areas at the DNA strand. And what happens with each copy, you might notice, we lose a little bit of the DNA strand from both ends. This is because the very last nucleotides of the DNA cannot be copied. So over time, it gets shorter, and eventually we reach this no more copies can be made signal, and this is how DNA stops copying of itself once it has been already copied too many times. And this is to protect so that mistakes do not accumulate over time. For example, cancer would be when the DNA copying goes uncontrolled. So there we have it, 3 nice examples of cell cycle regulation. And this is where we will wrap up this video on mitosis. Now that we have reviewed the concept of mitosis, so how all cells in the body, with the exception of the sex cells, sperm and egg, how all these cells get copied. So the next video that we will look at is going to look instead how the sperm and egg cells are copied. So we will talk about meiosis on that video.